Hello, everyone. I'm Miss Frizzle, and I think it's a perfect time for a field trip into the world of structural biology. And today we're going to follow our magic chameleon to the three NIH-funded National Cryo-Electron Microscopy Centers, where we will meet Ed Eng from the National Center for CryoEM Access and Training, NCCAT, Kirsten Wheeler from the Pacific Northwest CryoEM Center, PNCC, and Michael Schmidt from the Stanford SLAC CryoEM Center, S2C2. And they will briefly show us the resources we make available at no cost to the research community. Then Michael will introduce our speakers, Rakhi, Rajan, and Chhandosee Ganguly at the University of Oklahoma. And they will take us on a journey deep inside of an intriguing biomolecule. If you enjoy today's field trip, you can join us again every month with center users who generously share the tips and tricks that have enabled them to make use of CryoEM in their research. This bus departs usually the last Thursday of each month, but we'll actually skip November because of the Thanksgiving holiday. We'll be back on December 12th. And Alex Vecchio from the University of Buffalo will take us on a trip to learn about cryo-EM and fiducial marks as fail-safes for structural studies of very small membrane proteins. And the registration link for that talk is already available on the events page at cryoemcenters.org, where you can also find recordings of our past talks. So one final rule before we leave the classroom today, please send questions using the Q&A feature. And you can send those questions using the Q&A box at any time. Mike will moderate Q&A at the end of the talk, but send those questions in while you have them. So our first stop today is a visit to see Ed at NCCAT. So seatbelts, everyone. Let's go. Ed, take us away.
Okay. Hi, I'm coming from NCCAT. We offer high-end data collection, screening, and training access. Something that we have just commissioned is our Glacios G2. That's our second Glacios, and it's ready for users. That will augment not just our training categories, but screen categories. If you missed our workshop earlier in the month on SBT LabTech's blot-free vitrification device Chameleon, please do apply for our Chameleon Access category. Also, on the terms of training, next week we'll have our MicroAD short course. So if you missed that, don't worry, all the information will be online and how to apply to NCCAT at nccat.nysbc.org. See you soon.
Thank you, Ed. Now, let's go visit PNCC. Kirsten, let us know what's happening.
Perfect. Hi, my name is Kirsten Wheeler. I'm the administrative coordinator for Pacific Northwest CryoEM Center. PNCC has two different types of proposals. The first is our screening access proposal, which allows up to 2 users to come on site for up to 5 days of sample preparation and screening with staff. The proposal is aimed at low resource institutions. The second proposal type is general access, which awards up to 480 hours of microscope time a year for 2 years. This time can be utilized for screening or data collection. Approved general access proposals are also eligible for our training workshops, which cover topics from sample preparation through to data processing. We recently added an EPU data collection workshop this fall. It was very great. We've learned many things from it, which is fantastic, and we are excited to have our users attend that more often. PNCC also has 6 microscopes. We have an Artico with a K3 camera for screening and 4 Krios instruments for data collection equipped with K3 and Falcon 4i cameras. 3 of our Krios are equipped with Katana energy filters, while the other 2 have Thermo Fisher energy filters. Our Krios 1 just completed its upgrades to Selectrix X. The newest member of our microscope family, however, is the Glacios 2, which will be utilized for both screening and data collection. It is not currently up yet, but we are looking forward to its utilization. For sample preparation, we also have the 2 VitraBots and a Leica GP2. If you're interested in working with PNCC, please go to pncc.labworks.org and apply today. Deadlines for proposals are at the end of each month.
Thank you, Kirsten. And my research tells me there is one more national center to visit. So Mike, let us know what's happening at S2C2.
Morning. My name is Mike Schmid, and I'm one of the co-directors of Stanford Select CryoEM Center. We have 4 Cryoses, all commissioned and all running at the same time. That's very good. And we're commissioning our own Glacios that'll be up soon. So we offer, as all the other centers do, we offer high resolution data collection, screening, and training. And Rakhi and Chhandosee have made use of all of our equipment and all of our training opportunities and screening opportunities. One of the interesting things we have is a users meeting where they offer their own tips to each other on data processing and image processing, which happens every week. And Chhandosee is one of the stars of that group. We've learned a lot from each other. So join us at S2C2. We'd love to include you in our family.
All right, I'll stop sharing and let Mike introduce our speakers.
We're honored today to have Rakhi, Rajan, and Chhandosee Ganguly to speak to us today about CRISPR. Rakhi got her PhD at Ohio State University and did a postdoc with Alfonso Mondragon in Northwestern. Alfonso was one of the very first users of our CryoEM Center, so she has a great heritage from that kind of experience. She is now an associate professor in the Department of Chemistry and Biochemistry and member of the Price Family Foundation Institute of Structural Biology at the University of Oklahoma, Norman. And Chhandosee is a graduate student with Rakhi. I believe she started in 2021, and one of her first projects at S2C2 was in specimen preparation and optimization. So I hope that we can learn from both of them. Rakhi, why don't you start? Thank you.
Okay, thank you. I want to start by thanking Mike and Christina for this opportunity. And I want to say we are newcomers to CryoEM. So everything we have achieved so far has happened through S2C2 and NCCAT. So today, in the first part, I will talk about structure mechanisms of CRISPR-Cas systems. And then in the second part, Chhandosee will tell us how we resolve some of our issue with the sample prep. So a brief introduction into CRISPR-Cas systems. So these are bacterial and archaeal immune system. So in this cartoon, we can see this bacterial cell being infected with a virus, which is injecting its genetic material into the bacterial cell. A piece of this genetic material is excised and inserted sequence specifically into this CRISPR locus, which is later converted into specific RNAs called CRISPR RNA. And the CRISPR RNA then binds with specific proteins called Cas proteins, and they will bind and form a surveillance complex or effector complex, which will look for sequence complementarity with the invading DNA upon secondary infection. And once the complementarity is established, the Cas proteins will degrade the invading DNA and offer protection to the bacterium. So there are many different flavors of CRISPR-Cas systems. Right now, there are around 7 types, and that's based on the type of protein and the type of DNA target they are cleaving. And today we will focus on 2 of them, types 2, the signature protein being Cas9, and type 5, the signature protein being Cas12a. Okay, so now, why did CRISPR become such a big success in gene editing? And that's because of the ability to use RNA to target any DNA in a genomic context in any organism. And in this example, we are showing Cas9 targeting a DNA, and all it needs is 20 nucleotides of complementarity with the DNA along with a PAM sequence. So the PAM is needed to tell Cas9 whether it's an invader or a self-DNA. So once it locates the target, then Cas9 can introduce double-stranded break, which can be repaired either by non-homologous end joining or homologous recombination. So this gives the opportunity to create both knockouts and knock-ins using the Cas9 system. And recently, this system has been used for gene therapy. So we have Casgevy approved for treating blood disorders like sickle cell disease and beta thalassemia, and it has been approved both in UK and USA for use. But they have their own problems, and one of the main problem is off-target DNA cleavage. And as shown in this cartoon here, if we use our Cas9 RNA complex to target our genomic DNA, in addition to establishing a complementarity with the 20 nucleotide region, which we are interested in, it can also target and cleave regions which are partially complementary to the guide region of the CRISPR RNA. And as we can imagine, this will cause unintended DNA cleavages with undesired genome outcomes, and this increases actually the cost of CRISPR-Cas9 technology. So one of the things we are interested in the lab is actually understand the molecular mechanisms of how CRISPR proteins interact with the nucleic acid and see whether some of these mechanisms can be used to create safer genome applications. And today we will talk about one of the approaches we use in the lab, which is modifying the conformational change of proteins to remove off-target cleavage. And we will show two examples of Cas9 and Cas2A. So before I go into the details, I wanted to talk about a schematic introducing the different components of the Cas9 protein. So it is a bilobe protein, so it has a recognition lobe which binds to the RNA here, and then we have a nucleus lobe which houses the two endonucleases, RAUSI and HNH. These two lobes are connected by this conserved arginine-rich helix, which is called bridge helix, and this is the topic of our presentation today. And here in this R loop, we can see that the 20 nucleotide complementarity established with the target DNA. And once this is established, then the RAUSI and HNH can independently cleave the DNA, introducing double-stranded breaks. So as I said, one of the problem is off-target cleavage, and what is here in this example, we see that there is no complementarity between the RNA and DNA, and I would refer to this as mismatches in my talk. And several labs are using different approaches to remove the off-target cleavage, maybe by reducing non-specific interaction between protein and DNA, maybe reducing the length of the RNA to improve specificity. So there are multiple and directed evolution approaches. So there are a combination of things people are doing in the lab. But we focused on this bridge helix because we wanted to develop a method which we can translate between different Cas proteins and develop like a toolbox of different high fidelity Cas proteins. So when we started this project, we saw that if we look at the bridge helix of apoprotein and compare it to structures where it is bound to RNA or RNA and DNA, we see a loop to helical transformation in the bridge helix. And interestingly, this conformational change region is very close proximity to the RNA seed region and also the target DNA where the targeting is happening. In addition, it is also closely related to the endonuclease domains, which are involved in DNA cleavage. So we asked ourselves, can we interrupt this loop to helical conformation, maybe by introducing prolins, because prolins are helix breakers, and see how this will change the activity of the protein. So we made this, so even though the loop is of 4 amino acids, we changed 2 at this time, so L64P and K65P, and I will refer to this variant as Cas9 2-Pro in my talk. So this is the in vitro DNA cleavage assay, which we did with the wild type and our 2-Pro variant. As you can see here, so in vitro cleavage, we are providing a plasmid where we have this complementary region to the sgRNA guide, and then we are checking how easily the protein can cleave the DNA. So in the matched DNA, we have complete complementarity between the RNA and the DNA, and here we see that we have comparable cleavage for both these proteins. But when we introduce mismatches, so here we have mismatch at position 3 down the PAM, position 5 down the PAM, and position 7 down the PAM, you can really see that there is difference in how the wild-type Cas9 and the 2-Pro variant is cleaving the DNA. We see that 2-Pro is really sensitive to mismatches and the cleavage activity is reduced considerably. We followed up with this cell-based gene editing assay, which was done by Dr. Amrani in Sontemer lab. And this is testing one of the gene targets, DTS7. And again, we see that we have comparable on-target activity, but compared to the wild-type protein, we have much reduced off-target activity. So we established that this interfering with this loop-to helical conformation can reduce off-target cleavage. So the next thing we wanted to know, okay, why is this happening? How is this bridge helix helping the protein distinguish mismatches? And one of the things we noticed is that if we compare this gel, so this is how the protein is cleaving a mismatch at 5. Again, we are giving it a plasmid and checking how it cleaves either one strand or 2 strands of the DNA. So we see that we have an accumulation of the nick DNA in case of our 2-Pro protein, when the formation of linear product is much reduced in 2-Pro compared to what we see in the wild type. And as I showed you earlier, bridge helix is in very close contact to these RAWC and HNH domains. So we asked a question where the bridge helix mutation is affecting the activity of one of these domains. Previous literature has shown that even though RAWC is dependent on HNH for activation, because HNH prevents the accessibility of RAWC to the DNA, these both nucleases can independently cleave each strand of the DNA. So we asked which one of these domains are affected by the bridge helix mutation when we provide the protein with mismatch-containing DNA. And towards this, we use time course DNA cleavage assay. Again, we are monitoring the plasmid cleavage. So if you have cleavage by one domain, we see nick product and nicked intermediate. And if you have both cleavage, we get a linear product. So this is matched DNA or on-target DNA, where there is complete complementarity between the RNA guide and the DNA. We see that both the proteins are producing linear efficiently, even though for wild-type protein, we get almost 100% cleavage. Our 2-Pro is around 10% accumulation of nick DNA we still see. So again, a little lower efficient 2-Pro compared to wild-type, but still this is comparable with getting an efficient on-target cleavage. Now, if we compare the mismatches, so this top section have data for mismatch 5. Again, we see interesting difference. The wild-type protein has no distinction between the matched and mismatched. It cleaves it almost 100%, while our protein has only 30% cleavage, linearization, and we see accumulation of nick product at 50%. For MM18, which is another mismatch, which is very far away from the PAM, we see, again, wild-type completely linearizing the DNA, but our prolin mutant is accumulating around 30% of nick DNA. So now with all this data, we collaborated with Dr. Chin at USC, and we created a kinetic model, which is shown here. So here what we are showing is, so we are providing a supercoiled DNA, and to reach the final product of linear DNA, the protein can take 2 pathways which are independent of each other. So in this first pathway, we call TS pathway. The nicking is done by HNH with a rate constant of K1 HNH. And the second cut for linearization is done by RAWC with a rate constant of K2 RAWC. Similarly, for the other pathway, NTS pathway, the first cleavage is by RAWC with K1 RAWC rate constant, and the second one by HNH with K2 HNH. So these are 2 independent or parallel pathways which the protein can take to linearize the DNA. So we used our data. So we have the amount of supercoiled DNA, we have the amount of nick DNA, and we have the amount of linear DNA from our experiments. And we did some kinetic model fitting to get these 4 rate constants. And these are the rate constants for matched or on-target DNA. So what we see is the protein, wild-type protein, is mostly using this top pathway because we have higher rate constants for both these cleavage activities towards linearization. We do still have a small population using this pathway towards linearization. Now, if we compare the 2-pro variant, again, we see that the top pathway or TS pathway is what is most prominently used for linearization, but the rate constants have around 4-fold difference between the wild type and the 2-pro. And so we can conclude that for matched DNA, both of these proteins are using this TS pathway for linearization. Now, when we compare our data for MM5, we start to see interesting differences. The wild-type protein is still using the top pathway to linearize the DNA. Its rate constant has gone down a little compared to matched DNA, but still mostly it is using this top pathway. But if we come to our mutant, we see that the top pathway, which we believe is the most efficient way to linearize the DNA, is completely shut down because this K2 Rousey is taking a value of 0. But the protein is somehow trying to adjust for that problem by using a small part of this second pathway to linearize the DNA. Now, when we compare the next mismatch, which is MM18, we see another interesting feature because in this case, the TS pathway is completely active for both the proteins, but then the NTS pathway is shut down for our 2-pro variant. So this is a table summarizing the efficiency of pathway usage by these proteins for all these different DNA substrate. And this we calculated by taking a ratio of K2 over K1, because since K2 is a linearization, the efficiency of linearization we can get from this ratio. So if we compare the wild type protein for all the type of DNA, it is using the target strand pathway. If we compare the 2-pro and matched, it's using target strand to cleave the matched DNA. But when it comes to the mismatched DNA, it is completely shutting down the TS pathway, but using the NTS pathway for linearization. And we see that in the mismatch 18, we see the opposite. We see it using TS pathway for linearization, but nothing from NTS pathway. So what does this all mean? So it means that we cannot generalize which endonuclease domain is impacted by mutation in bridge helix, and it actually depends on the position of mismatch. So it's like an allosteric control bridge helix is offering to HNH and Rousey, and the control mechanism depends on the position of the mismatch. Now, we also introduced alanine to the bridge helix because the idea is if proline is inhibiting cleavage, then alanine should bring it back because alanine has a high propensity to be in a helix. And this, I'm not showing a lot of data, but that's what we see here. We see that when we introduce alanine and try cleavage with a mismatch 5 DNA, it starts behaving very closer to the wild type protein. And we still see a lot greater accumulation with our 2-pro variant. So the conclusions, bridge helix is, for this part of the talk, bridge helix is allosterically controlling the independent strand cleavage activities of HNH and Rousey. And the allosteric control mechanism depends on the position of the mismatch. And we can tap this mechanism to create high fidelity tools. So the next question, and some of the questions we are currently addressing is this mechanism conserved in other orthologs of Cas9 and also in other enzymes which possess bridge helix? And what is the mechanism of allosteric control by the bridge helix? And we are hoping to use CryoEM to answer some of the questions. So now looking at Castrol Way, this is a cartoon again representing the domain architecture. We see that we have similar bilobed architecture, having a rec lobe and a nuke lobe connected by a bridge helix. But there are some interesting differences. This protein uses only one RNA, the CRISPR RNA. And also this protein is much smaller, so people like it for delivery options. Another interesting difference is protein uses only one endonuclease domain, Rousey, to make cleavage of the DNA. And it has like a sequential cleavage since there is only one endonuclease. So they take an order in which the cleavage happens. So we compared the bridge helix of Castrol Way with that of Cas9. Right away, we can see that the bridge helix is much shorter in Castrol Way, but it is still maintaining some of the similar interactions with the RNA-DNA hybrid. And comparing some of the structures available in the literature, we could see there is some helical transformation in binary and ternary structures. So we again did the same thing. We introduced prolines at this time at positions 969 and 970. We used a Francisella novicida ortholog, and we call this as Castrol Way KD2P. And this is again plasmid cleavage showing how the two variant behaves compared to the wild type. So we see that again, when you don't have a mismatch, both of the proteins have almost equal efficiency, but this KD2P variant is much impacted in mismatch cleavage. In fact, in this variant, we were able to test each and every position for this protein, and we can see that linearization is much reduced for most of the mismatch positions. And another interesting thing I wanted to say is there is this activity for Cas12a called trans-DNA cleavage, and that is the basis for all of these molecular diagnostic tools using Cas12a. And here what happens is after Cas12a does this RNA-dependent DNA cleavage, which is also called cis cleavage, the one I just explained, the Rousey active site stays open, and it is able to take in single-stranded DNA and degrade it non-specifically. So while this might be a good activity for a molecular diagnostic tool, we can imagine this might not be advantageous for a gene editing application. So we checked whether bridge helix contributes to transcleavage, and we were very excited to see that the bridge helix variation removed the transcleavage, as you can see here. So basically, this shows that the bridge helix is somehow controlling both the cis and transcleavage of this protein. So now we had some interesting results on bridge helix and controlling DNA cleavage. We wanted to get more insights on what is happening and what is the molecular mechanisms behind that. And we decided we want to use CryoEM. But at that time, when we started this, we didn't have resources in our university to do the CryoEM. So most of this is done. So again, a group of faculty and facility manager in our department, there are Dr. West, me, and Dr. Bond, and Dr. Thomas. We initiated the CryoEM single particle analysis in our department, mostly through CryoEM facilities and our collaborators. So we started by acquiring a Leica plunger because this gave us the ability to freeze our grids and optimize our samples as we liked, and this gave us more flexibility. And then for the rest, for access for microscope and also for training, we mostly depended on national facilities. So we have been using S2C2 since 2022 and NCCAT since 2023. So just a little bit on how we get to the stage of preparing a grid. So we purify our complex over a size exclusion column, and we usually see 3 peaks and check them on a native gel, and we realized peak 2 is what we want. And we take the peak 2 and put it on a grid, and then we freeze it. And again, we had many issues trying to get the particles on the grid, and Chhandosee is going to explain a lot on how we overcome those challenges. But again, when we started, we couldn't see our particles in the holes, and then we figured out that graphene oxide coated grids are helping us get the particles uniformly distributed in the holes. So the best thing is we optimize this in NCCAT, that graphene oxide is the way to go. And then we use that to go to SLAC to collect our data. So this is how we used our, like mostly we have 1 to 2 month waiting period between these two facilities. So this is how we made sure we have continuity, take one thing from one facility and take it to the other facility. And that has really helped us progress in our efforts. So we collected a data set at SLAC, and we had 1.6 million particles, and we used 2D classification, and we used 2D classification as a way to clean up our particles, and we ended up with 400K particles. And these are some representative 2D classes for Cas12a shown here. And interestingly, we also get 2D classes for our graphene oxide, which we can reject during this iterative purification process, I may say, of this data processing. Okay, so we used 400K particles, made like an ab initio map, and we see that it has this crab claw shaped feature, which was seen for the wild type protein, and we were able to fit the wild type protein into our model. And after homogeneous refinement, we have a resolution of 2.96 angstrom shown here. But then we went ahead and did 3D variability analysis, and based on that, we got 6 classes, which is shown here. And the resolutions range from around 3.2 to around 8 angstrom. And one of the interesting fact here is most of them have the same percentage of particles, showing that there is no like optimization or stabilization of one intermediate conformation. The protein is actually transitioning between these uniformly equally in our solution. So we decided we will further go ahead with these 5 models and 5 maps and make models. This one, because it was 8 angstrom, we didn't proceed further. So this is another example showing the local resolution of the different maps. So we have around 3.5 to 4 for most of the regions of our map, and we have made models to fit this map. So this again is showing the variability between the different classes we saw. So here is a structure of our class 4 with the different domains color-coded and as shown here. So what we see is most of the variability is coming from the Rec1 and Rec2 domain as shown here. We see that these are undergoing large movements in between the different classes. So now we wanted to see how can we place them in the order of their DNA cleavage catalysis. And we referred to wild type structures which were available on the database and compared our structures to that. And currently we have this as our cycle of DNA cleavage. And what we see is in this state 1, we don't see much density for DNA. So DNA is supposed to bind here in between this PI domain and this reg domain. And we do not see much density for DNA. So we put that as our starting state. And we also see that this Rec2 and the nuke domain are actually shut in this position as if it is occluding anything binding in between. But when we go to state 2, we see that the Rec2 domain is starting to move away from the nuke domain. And in state 3, the Rec2 is invisible. And in state 4, we see it slightly coming back. And in state 5, it is completely present. But the orientation of this rec domain is different from this rec domain. And again, we have used previous studies in wild-type protein, which has used FRET studies to assign different states for this protein. So one thing also we noticed is even though we get all these different states compared to the wild type, we are actually not sampling that state where everything is set up and ready for DNA cleavage or the pre-catalytic stage. So we assume that introducing this mutation in the bridge helix is somehow reducing the sampling of this protein to reach that DNA cleavage competent state. So that is where we are with CryoEM. Currently, we are doing more analysis to see, to make a full story on what bridge helix is doing. But then in the meantime, we also looked at alanine substitutions for Cas12a. Again, similar to the previous Cas9, if bridge helix helicity is important, when we put it, when we substitute the prolines with alanine, we should really see that the protein is becoming more active and able to tolerate mismatches. So this is the data for matched DNA or on-target DNA. And you can really see that this wild type and the KD2A, where we have 2 alanines, they are very efficient in cleaving. And in fact, if we compare the rate constants, we see that the alanine substitution is twice as effective as the wild type in linearizing the DNA. And we see that our protein has much lower efficiency in linearizing, but with given enough time, it catches up and does a good job of linearizing the DNA when the DNA is matched or on target. Now we tested this with 2 mismatches. Here we selected MM2 and MM13. We selected this because previous experiments have shown MM2 is not discriminated well by KD2P, but MM13 is clearly discriminated by this protein. So if you see here, again, the wild type and KD2A are linearizing the DNA very effectively. With higher rate constants, but then KD2P is lower, but still reaching almost 80% efficiency of DNA cleavage. So we can say there is 20% discrimination here compared to wild type and KD2A. But when we compare the mismatch 13, we see that the wild type has some discrimination. In fact, it is linearizing only 60% of our substrate, but the KD2A has completely lost ability to do any discrimination, and it is cleaving the DNA at 100%. And very interestingly, our protein 2-pro KD2P cannot linearize this mismatch at all, and we have 0% linearization. So another thing we can find from here is that the discrimination is occurring at the second stage DNA cleavage, because in all of these we do see production of nicked intermediate. So the discrimination is happening at the second step of DNA cleavage. So for conclusion, we have shown that loop to helical transition and stability of helical structure contribute to DNA cleavage and also sensing mismatches in Castrol way. And it looks like nature has evolved the bridge helix of Castrol way and also Cas9 in a way that the amino acids there are giving it some flexibility to accommodate DNA mismatches. And maybe that's a good immune system to cut some of these viral mutations which are evolving. So when we use such proteins for gene editing, then we need to fine tune the amino acids in this bridge helix so those flexibility of cutting mismatches is reduced. So the open questions which we are currently addressing is how is it sensing mismatches in Castrol way, and what is the difference in sensing this DNA mismatch in Cas9 and Castrol way using the bridge helix. So I am happy to report that right now we have a Chandra microscope in our department right down one floor, and we are all very excited with this development because this is really going to accelerate our CryoEM efforts. And I think our training through the national facilities had been an important drive to reach at this stage. So that's all I have. I want to then acknowledge all our collaborators and national facilities. So Dr. Peter Chin, we collaborate at University of Southern California, and these are his lab members who contributed to the work which I presented today. Dr. Jin Lu, she is at University of North Texas Health Science Center, and she helps us with molecular dynamic simulations. I didn't show the data, but we have used simulations to understand the mechanism side by side. And at SLAC, I want to say special thanks to Nathan and Dr. Bachu because they have been very instrumental in helping us reach here. And at NCCAT, I have all these members to thank, Christina and Ed and other members who trained us while we were there. Martin Lawrence and lab members at Montana State University, Alfonso, Amy, and Valerie at Northwestern University. And OU core facilities, Dr. Len Thomas, so he worked side by side with us on CryoEM. Dr. Bone is in the protein production and characterization core on preparing the proteins. And these are the current funding for this project. And finally, I want to thank my lab members. So these are the previous people in my lab who contributed to the work. So Dr. Yogesha, Keshavan, and Ramya, and also 2 graduate students, Priya and Sydney, and several undergraduates who helped us reach at this point. And this is my current lab member. So Chhandosee is the one who spearheaded CryoEM in my lab. Lindsay Martin is the one who purified all these complexes, and some of these proteins are not very friendly. And she also made these complexes and did all this careful kinetic analysis. So with that, I will stop and don't know whether we are taking questions now or later.
Thank you. There are no open questions now, but I'll have one at least for you at the end. Okay, so you can go to Chhandosee.
Is my screen visible?
Yes, it is.
Okay.
Great.
So, good afternoon, everyone. Today, I will be sharing our journey towards achieving an even single particle distribution of our complexes throughout this process. When we first began, we encountered a range of multiple challenges, including, oh, let me just get my pointer, please.
And Chhandosee, we don't have your video, and that's fine if you intended, but if you want us to see you, you can show that too. I'll let you decide.
Yeah. So, yeah, so we encountered a range of challenges, including I don't know why the slide is not going. including empty holes, particles sticking to the carbon, and aggregation of complexes. So in order to get through all these challenges, we received immense help from the national facilities, especially S2C2 and NCCAT, and then we were finally able to achieve what we wanted to as a structure. So I'll illustrate some of the example images of what problems we were encountering. First of all, we had mostly empty holes with no particles in the holes of the grids, and mostly the particles would stick to the carbon. And in some other cases, we will have large aggregation of our complexes on the grids. So let me go ahead and explain a little bit about our protein purification method. So we make our complexes in vitro, and then we purify them using size exclusion chromatography. In order to verify the presence of the components in our complexes, we run our fractions on the native gels. And here you can see that we use cybergreen stain to confirm the presence of nucleic acid first, followed by Comasi blue stain to confirm the presence of protein. Additionally, we also run the denaturing gel to make sure that all of our components are present in the fractions we are using. After confirmation, we take the peak fraction and we directly apply it on our grids. The first parameter which we worked on optimization was for the grid preparation. Initially, when we were preparing grids, we had access only to the Pelco glow discharger for glow discharging our grids and Leica EMGP2 for plunge freezing our grids. And these were the following parameters which we were following. However, they were not giving us any satisfactory results for our samples. In June 2023, when we went to NCCAT and we got a lot of access to the resources and invaluable guidance from the experts, we were able to optimize our parameters, and we were lucky that we got access to Solaris as well as the Vetribot because these were the instruments which were mostly cited by the literature available in our field. And then we were able to optimize our preparation with a combination of Solaris with Vetribot as well as Pelco with Leica. And these are the parameters we are currently using. Then we tried different strategies to get our particles into the hole. The first thing we tried was to use different type of detergents. Here, there is an example of one of the detergents we use, Chapso. When we use Chapso, we were unable to achieve single particle distribution into our holes. However, the aggregation still persisted. We went ahead and we tried polylysine coated grids. In the polylysine coated grids, yes, there were few single particles which we were able to see in the holes, but there were a lot of aggregates too. And the number of single particles were insufficient for data collection. The third thing we tried was graphene coated grids. In the graphene coated grids, we didn't notice a lot of aggregates, but the particle density was extremely low to do any data collection or go ahead and determine a structure. Finally, we went ahead and we made graphene oxide coated grids. And when we made graphene oxide coated grids with the help of personnel at NCCAT, then we were able to finally see our single particles into our grid holes. And you can see from compared to the previous images, we have very good distribution in this grid, in this micrographs. The particles have been encircled in white circles. So we did all this optimization using our Cas9 complexes, and when we extended this to our Cas12a complexes, we could achieve the consistency of our single particle distribution. So graphene oxide really turned out to be a game changer in our case, and our samples love being inside the hole now. So let me briefly take you through the process of our graphene oxide preparation. So we usually use the quantifoil copper grids. with 300 mesh, which we first clean with chloroform, followed by glow discharge, an application of polyethylene amine coating, and then graphene oxide coating. And once the grids are properly dried, then we apply our sample and we freeze our grids. The protocol for preparation of this graphene oxide grids have been adopted from the two following literatures. Now we know that the formation of the monolayer graphene oxide has been a consistent problem in the field. So we went ahead and we wanted to check how much consistency can we achieve with our monolayers. So the data set which we collected for Cas12a, we randomly picked 100 micrographs and we did an analysis, and we found that there were 34% of the micrographs which had 6 diffraction spot indicating monolayer of graphene oxide, and then 22% of the micrographs had 12 diffraction spot indicating bilayer of graphene oxide, and there were few with multiple folds of graphene oxide giving multiple diffraction spots. But all this collectively were good for us to get us final structure. Later, to check the reproducibility of our graphene oxide coating, we evaluated our grids multiple times during screening through S2C2 and NC-CAT, and we could see that there was a good consistency in the uniform graphene oxide layer. And recently, we imaged some of our grids at our facility at OU using our new Tundra. And then we could see the similar uniform layering of graphene oxide. These grids were imaged with no sample on them at multiple magnifications. As you can see first, this is at the square magnification where we don't see multiple layerings on this grid square. The same grid was taken to the whole mag, and in the whole mag, these grids, these holes have good and uniform graphene oxide layer. These are some of the examples where you see that there is no layering, like these holes. And this is one of the example where there were multiple folds of graphene oxide, but we still don't see multiple layers of graphene oxide. And then in these regions of carbon, mostly you can see the folds are there and not into the holes. So that's good for us because we want them to be uniform mainly into the holes. And this is the image which was taken at the exposure magnification, and that also looks pretty clean without folds. So in conclusion, from the beginning or the before scenario when we started, we had a lot of aggregated complexes or literally no particles in our holes. And finally, these were the 3 major contributing factors which helped us to achieve even distribution of single particle throughout in our grid holes. At the end, I would like to thank everyone at S2C2 and NC-CAT for letting me learn CryoEM. And if I could have achieved any structure today, it's just because of all of your training and teaching. And a big thank you to all of you. And at the end, happy Halloween.
Thank you.
You would hear a great round of applause if our audience could let their applause be known. That was really fantastic. Thank you.
Good. Okay, Rakhi, I have a question about the specificity of matched versus mismatched DNA. And in your experiments, you have either purely matched or purely mismatched DNA. And in the real world, there would be an ocean of DNA of all kinds, with varying from no matches at all to perfect matches with mismatched in between. And the rate constants you see for say one mismatch versus wild type, or sorry, not wild type, but perfectly matched DNA. That's a pretty reasonable difference, but not a big difference. But if you had the presence of all kinds of DNA, would perfectly matched DNA win out more often just because there are so many more chances for mismatches to be discarded and the rate go down. I'm just saying, in the presence of all the different kinds of DNA that the complex could see, is this mismatch discrimination good enough? You're muted.
Can you hear me now?
Yes.
Okay, I think, okay, so great question. So in general, what people have seen is some of the rate constants we determine by in vitro cleavage is translated to what we see in cell-based assays. So in fact, people have done these type of cleavage with the whole cell, and they have taken DNA and done some measurements. So we could see still some of these facts holding true when we go from the test tube to the cells. But one of the important factors that the accessibility of DNA, I feel, is a major player in how Cas9 and Cas12a are able to target. So open chromatins may be as easily targeted, and some of the rules we are seeing in the test tube might easily translate to those type of open chromatin regions. But maybe if a region is not accessible, I cannot say whether we can truly say this will translate to what we see in the test tube. So did I answer that question?
Yes, thank you. Yeah, protected versus non-protected DNA is, that's a different, that's another issue, of course. And
Yeah, I have a question for Chhandosee, if I can pipe in with a question of my own. So I know a lot of the optimization of these graphene oxide grid preps is like very dependent on the exact reagents. So I'm sort of curious, after you did some optimization here at NCCAT, when you got home, how many iterations did it take you to get the protocol that gave you consistent results back at home, like with potentially a different bottle of the graphene oxide flakes and that sort of variability?
I think that was very interesting that it was not very difficult. And I saw the definitive consistency because some of the images I showed were taken at NCCAD when we made the grids there. But the data which we collected was the grids which were made at home and which I sent to S2C2. So most of the data which is sent to, like the pictures which were taken at S2C2 were prepared by the graphene oxide bottle at OU. And the NCCAT ones are prepared by the graphene oxide bottle at NCCAT, and they are pretty much consistent. We have even seen consistency in hands, like someone else in my lab prepared, and they also were able to achieve the monolayer of graphene oxide.
Awesome. That is encouraging to hear.
yeah, reproducibility is key. So here is a question. Sorry if I missed this, but what exactly helped sort out the aggregation problem? So you mentioned you mentioned the distribution and sticking to holes and not. So what do you think solved the aggregation problem particularly?
We were optimizing multiple things at the same time. So I would say it was a combination of things, but definitely our complex purification, and we started putting it on the grid right away. And we didn't do any kind of incubation because sometimes we would do incubation and it led to formation of aggregates. We have fortunately never seen any aggregation on graphene oxide grids. And other than that, I think we optimized a little bit in our buffers also. We added a little bit of glycerol, which was not there previously. That helped with the aggregation problem too.
And here's another question. Did you use specific reagents during the protein prep? So that's just making specifically detergents in the protein prep itself.
Not the protein itself, but during the complex purification, we used Chapso. And that was one of my results. And we didn't see any improvement in the particle distribution, or the aggregation was still persistent there after. We also used another cationic detergent. It's called CTAB. And that also we didn't see any improvement.
And then I have another question for Rakhi about and Chhandosee about your choice of when you take a peak from the size exclusion column, and another peak might be near it, that either preceding it or following it. Is it best in terms of the number of the concentration would be highest at the peak, but the purity, does the purity depend on whether you take the peak or a little bit on the other side of the peak from another peak, just to keep it as far away from some other component as possible? Or do you see that, okay, the peak is really very pure, that and nothing else, in terms of maybe in terms of classification later? I don't know.
So when we make grids, most of the time we are sending multiple grids, right? So we take different fractions. So we take the absolute peak fraction and we'll take one ahead and one afterwards to check. And we definitely make sure that in the gel, they look similar. So they don't have like different components in there, but we do different fractions because the concentration, we use nanodrop to quantify them. But we cannot absolutely say that that is the concentration due to presence of RNA DNA giving higher absorbance even at 280.
But I also want to add, even with all those precautions, we got a mix of things, right? So we do as careful as we can, picking at the tip of the peak and taking very small volume of each fraction, like 50 or 100 microliters. So we are hopefully getting the purest homogeneous protein complex as we can, but still we ended up seeing different conformation. And in one of the structures, we don't even know whether the DNA is there. So again, we don't know whether the protein is actually a mix in the peak or is it falling apart during the grid preparation? We don't know. And we also had one class which we didn't model build. We don't know what is in there. So the bottom line is we do every practice we can to ensure homogeneity, but at the end, we can only do so much to control that.
Great. And does your analysis of all these different kinds of helix and other mutations indicate that maybe what people have to do is kind of tailor the kind of Cas that they're using and the kind of bridge helix they want to the particular piece of DNA that they want to perfectly match? And is it going to be a very customized process, or is there some generalizations that will arise out of this?
Actually, that's a really interesting thought. I never thought about it before, but it can make a difference, right? Because we are looking at only 2 amino acids, but it is in the midst of other amino acids and other side chains, which will give different type of interactions to the DNA backbone. So if you're going to do a comprehensive analysis of what is the amino acid near the in the vicinity of this helical transitioning region and how they contribute to the interaction, then I think we can achieve something like a customized bridge helix to different type of DNA sequences.
And then again, here's a question. Did you have to remove the glycerol while you were putting the sample on the grid? Or
no. So my buffer had 1% glycerol. And I think in literature, we have seen until 5% we can make grids without removing the glycerol. So we never removed the glycerol.
Well, I think we got to the questions that came in, and I'll have us finish up on time today. So thank you, Rakhi and Chhandosee, for sharing your journey. And thank you to everyone who joined us on our CryoEM field trip today. We wish all of you well in your own cryo-EM journeys, and the national centers are here to support your science. So come and visit us in person on whichever bus gets you here, and let us know how we can help. Thank you.
Thank you.